Welcome to The Explainer. Today, we're looking at a fundamental biological process that customizes the building blocks of life. It's this whole other layer of information beyond our DNA, a kind of sugar code that gives proteins their specific powers. To get us started, let's kick off with a puzzle. Think about the coldest parts of our planet. We all know water freezes, so how in the world do some fish manage to survive in Arctic and Antarctic waters that are literally freezing solid? Well, the answer is basically a biological superpower. In their blood, they've got these incredible molecules called antifreeze glycoproteins. These are just proteins that are coated in very specific sugar chains, and they act like a natural antifreeze. They literally get in the way of ice crystals, stopping them from forming and growing, which would otherwise be deadly. So this incredible adaptation in fish, it's actually a super dramatic example of a process that is happening inside of you, in your own cells right this second. It's called glycosylation, and it is absolutely essential for life as we know it.
Okay, so what is glycosylation exactly? Well, at its core, it's the process where the cell's machinery takes these complex sugar chains, they're also known as glycans, and attaches them to other molecules, like proteins. And this isn't random, it's an incredibly precise and vital modification.
A really great way to picture this is to think of a hidden architect working inside our cells. This architect takes a standard, freshly made protein, sort of the basic building block, and adds these really intricate sugar structures to it, turning it into a highly specialized tool designed for one very specific job. And, just like any architect, this one has its own set of blueprints. To even begin to understand the incredibly complex structures it builds, scientists needed a way to draw them, a kind of universal language to describe all these sugar chains.
And this is how they solve that problem. It's called the symbol nomenclature for glycans, or SNFG. It's just so elegant. Each one of these shapes and colors represents a specific sugar. So for example, those green circles you see are a sugar called mannose, and the blue squares are another common one called GlcNAc. It turns a super complicated chemical formula into a clear, readable blueprint. It's brilliant.
Okay, so we know what the architect is building, but where's the workshop? Let's take a tour of the cellular factory where all these proteins get their sugar coats. So, the factory is split into two main departments. The whole process kicks off in the endoplasmic reticulum, or the ER, you can see that up at the top, and then everything moves to the Golgi apparatus for all the finishing touches. It is an incredibly sophisticated assembly line. Let's zoom right in on the assembly line for one of the major product lines, something called N-glycans. The whole thing begins right there on the factory floor, the ER. Here, a standard prefabricated sugar tree is built piece by piece on this special lipid carrier molecule called Dolichol, which kind of acts like a temporary scaffold. And this whole process is just wild. It happens in 4 key steps. First, the foundation. A base of 7 sugars gets built on the outside of the ER. Second, and this is an amazing move, it's called the flip. The entire structure is moved across the membrane to the inside. Third, completion. The sugar tree is all finished up, forming a 14-sugar block. And finally, the big moment, transfer. This whole pre-made block is attached all in one go to a brand new protein. But hold on, the job's not done yet. The protein, now with its starter sugar chain, moves on to the Golgi. You can think of the Golgi as the customization shop and the shipping department all rolled into one. Here, that initial sugar block gets trimmed down and new sugars are added, tailoring the glycan for its final specific job. So with this whole assembly line process, you can imagine the stunning variety of things our architect can build. Now that we've seen the factory, let's go check out the gallery of its 2 main architectural styles. Okay, so the big difference here really comes down to strategy. You've got N-linked glycosylation, the one we just saw, which starts with that big prefabricated block in the ER. But then there's O-linked glycosylation, and that's more like a custom piece-by-piece build. It happens mostly in the Golgi, where sugars get added one at a time directly onto the protein. And wow, when you see the results side by side, the difference is just obvious, isn't it? On the left, you've got those N-glycans with their complex tree-like branches, and then on the right, you see a mucin protein, which is what makes mucus slippery. It's so heavily coated with O-linked sugars that it creates this incredibly dense bottle brush structure. The protein backbone is almost completely hidden.
All right, we've seen the what, And we've seen the how, but now for the most important part, the so what. Why does this hidden sugar code matter so much for biology and for you and me?
Let's start with a perfect example you carry around with you every single day: your blood type. The crucial difference between type A, B, and O blood, it's not in the protein itself. It's just the final sugar that gets added to the end of a glycan chain on your red blood cells. And that one tiny sugar difference is literally a matter of life and death when it comes to blood transfusions. It's amazing. These sugar coatings also act as bodyguards for proteins. They add bulk and structure, which helps the protein fold into its correct 3D shape and stay that way. This dense sugar coat also forms a physical shield, protecting the protein from enzymes that would otherwise just chop it up and destroy it. So they're not just shields, they're also a kind of cellular GPS. Specific sugar patterns act like shipping labels, telling the cell exactly where that protein needs to go to do its job. And getting that glycosylation right is a critical quality control step to make sure the protein folds correctly in the first place. And listen to this quote, because it really makes the stakes crystal clear. If you stop glycosylation, you don't just get slightly less effective proteins. No, you get useless, misfolded clumps of protein that can't do their jobs at all and can even become toxic to the cell. So when you put it all together, from the extremes of Arctic survival to something as fundamental as our own blood type, it's so clear that these sugar modifications are not just decorative fluff. They are a critical, information-rich layer of biology that is absolutely fundamental to how proteins work and how life survives. And that leaves us with a pretty fascinating thought to chew on. Our cells are covered in this incredibly complex sugar code, and we're really just starting to learn how to read it. So as our tools for reading and even writing this code get better, what new secrets of health, disease, and the fundamental rules of life are we going to unlock?